Welcome all attendees. We're gonna wait a few minutes for everyone to trickle in, and then we will kick things off. And I'll fill space with humor. Did y'all hear about the fortune-telling dwarf that escaped from prison? Yeah, it's a small medium at large. So we moved recently, and I think something happening in moving. I think my wife spilled glue on my rifle collection. Now she denies it, but I'm sticking to my guns. New town we're in, I saw online they have a zoo, and so I got all excited, took the kids to the zoo, and there was only one animal in it, just this little dog. It was a Shih Tzu. My wife says I should have known better. She says she told me, like she heard from a neighbor that it was like that. But my wife says there's two things wrong with me. One, I don't listen. And two, some second thing. But I mean, you know, wives have opinions. The other day she says to me, "You haven't been listening to a single word I've been saying, have you?" And I just thought, like, that's a strange way to start a conversation, you know? Anyway, okay, my humor's all spent. 2 minutes. I hope everybody showed up now. Shall we begin?
Let's go.
Why not?
Okay, welcome to our Biome Makers Chonex joint webinar on understanding, starting to unpack and comprehend variability and performance in biostimulants and biological products. I'm Scott McElveen. I am a technical sales rep here at Biome Makers. I would like to present Will Wright, Chief Operating Officer, Chonex. Will, would you say a few words of greeting?
Thanks, Scott. We're excited to be here. We've been working with Biome Makers for over 5 years now, and so it's great to be able to work together to get insights on how we all can improve soil health. So thanks for having us.
Our pleasure. And here we also have Dr. Jacob Parnell, the Vice Regent of Agronomy. He wanted me to just say agronomist, but our head of agronomy here in North America for Biome Makers. Dr. Jacob, would you say a few words?
Pleased to be here. Love all of the emphasis that's looking at the biology of soils now. It's kind of like we've reached our heyday now. People are finally paying attention to the biology of soils. So it's great to be here.
It is very difficult to argue that soil life and soil function and soil health are anything but essential anymore. Nevertheless, trying to manage soil biology in large-scale industrial agriculture presents some problems, one of which is predictability. So who is familiar with this sort of chart? Dr. Jacob, Will, are you familiar? Have you seen these in your time in this industry?
Yes.
This is a familiar sight, often referred to as the piano graph. And this results from doing a whole bunch of multi-site trials and simply measuring yield improvement or measuring metrics that we're used to measuring in agriculture in the past, not honing in on specific biologically relevant metrics to understand performance. And so these charts, typically, they have to skew to the right if you're going to have a commercialized product at all. I have just borrowed this. I did a Google search for piano chart of biological performance. I borrowed this, anonymized it. But a whole lot of any of these outcomes are going to be a whole lot of nothing. The difference, the change in performance, or sorry, the change in outcomes for the grower at the end of the day is a wash. It's just pure chance whether it goes up or down. Essentially nothing happens, nothing of note. Then you have the enough benefit that it was noticeable and it justified the purchase, 3x ROI. Then you've got another section that is the happy customer section. The guys in the justified purchase section, they're not writing home about it. they're probably going to try something else next year, perhaps, that would give them a chance of a better outcome. Happy customers, they're likely to come back. I mean, they saw a noticeable difference. They saw benefit. This is, you know, the core audience you're looking for. Then there are often some of these over here that are just, wow, Shazam. Is this a fluke? What happened here? If only we could understand what this was. I've spoken with companies who saw, say, 90 bushel increase, and it wasn't even that the control was that bad. And they just go, we can't even talk about this. We don't know what happened. It's obviously a fluke. It doesn't fit the pattern of everything else. But wow, these do happen. And then on the other end, you have the outcomes that are just get you raked over the coals. Again, in the other direction, what happened? Our product had the opposite effects that we intended. We need to, gosh, make sure this doesn't happen, because this is going to be potentially the most outspoken of the user base. And all of this chart all together, averaged out into a single metric, gives you plus 4 to 5 bushels per acre, which, how many times have we heard that? Get in line, join the club with everybody else, 4 to 5 bushels an acre. And what we're seeing here is variability as a liability. If you don't have a handle on how the product works, what's the mode of action, what are the factors that inform whether it's going to work in this or that scenario, you end up with a chart like this, and you got a better chance of a win than a loss, but you're rolling the dice a bit.
Well,
So,
and Scott, one important thing is this is a success case, right? This is something that does go to market.
yes,
And so this is not talking about just everything.
absolutely. The ones that don't skew right, like this one, never make it to market. They don't get commercialized. It's back to the drawing board.
Even though they might do something really awesome in those one places, you know. So yeah. So to me, it just boggles the mind.
Indeed. And so, in order to manage soil biology better with the products that we're developing, we have to think differently than we have in the past. I just want to illustrate why the degree of complexity in these sorts of products demands different thinking and different approaches to trials. So here we have some of our most common agricultural chemicals and fertilizers. We've got our azoxystrobin, DAP, imidacloprid, gypsum, glyphosate, chelated zinc, zinc oxide. These are very well-defined, distinct chemical compounds. And so you could, I don't know if this is going to help anyone but me, but if I were to express a simplified equation of performance, I might say active ingredient times conditions. And conditions is the huge umbrella that encompasses everything else: the climate, the environment, the soil, the skill of the applicator, adjuvants, and so on and so forth. If we move up a level of complexity to, say, plant extracts, seaweed extracts, protein hydrolysates, say hormones built to trigger a cascade of different behaviors and secondary metabolites in plants, immunity priming peptides or chitosans, then we'd say, okay, there's an array of bioactive ingredients and a set of combinatorial actions between those biologically active components times our condition catch-all umbrella. Level up again to living organisms. Here we have our Pseudomonas aeruginosa and some fungal spores. Well, now we've got the number of microorganisms squared, because there's crosstalk, they interact with one another, times their different number of secondary metabolites and hormones and precursors and aminos and so on that they can produce, times environment, times renewal. These can colonize, establish, live on into the future, and act later. All of this, I don't mean to bog us down with the equation, but I'm trying to illustrate how the complexity is jumping up by multiples as we climb into higher levels of biological activity, higher levels of biological, well, complexity, for lack of a better word. And so, Will, I would love for you to tell us about the product that we're going to be looking at today as a case study in order to place it for us on this ladder of complexity.
Yeah, thanks, Scott. Happy to talk about StrongSoil. So at Chonex, we've spent 6 years and a fair bit of money investing in creating StrongSoil. And StrongSoil is a concentration of the beneficial bacteria in layer hen manure. We have developed a biorefining process to remove the pathogens from that manure. Manure has been used for, as a fertilizer for millennia, but it is pretty inconsistent, and there is a challenge around pathogens, given that it is a waste product. So what we've done is we've taken that layer hen manure, we process it with black soldier flies, as well as a patented process to concentrate the beneficial biology from the layer hen manure, as well as from the black soldier flies. We then concentrate this down into a water dispersible granule that farmers can use dry. It ships dry. They can store it for 2 years. But when you need to increase the biology in soils, by mixing this WDG in water, you get a microbial bloom from that beneficial biology, and that then applied to fields is where we get our agronomic outcome. So that's the product that we've been working on for several years to develop. And it's exciting to be able to talk about the impacts that we have today. If you go to the next slide, Scott, just to be clear, kind of what is in StrongSoil? It is first and foremost a very complex and concentrated microbial biological fertilizer, essentially. So we have isolated within over multiple studies 8 highly functional bacterial groups that we consistently see in the product. Time and time again, and so that consistency is something we've worked hard to be able to deliver. This includes nitrogen cycling as well as chitinase producing groups. There is also a biostimulant aspect to this. So the black soldier fly larvae, when they're processing the manure, they shed their skins, which are loaded with chitin, their exoskeletons. And so by concentrating that, when we grind up the product through our processing, we capture that chitin in the product. So you also get that chitin when you're putting StrongSoil out in your fields. And then very importantly, because we are working with specialty crops, as well as leafy greens, et cetera, it is pathogen free. So we test every batch to make sure that there are no fecal coliforms, salmonella, anything like that. And so being able to provide all the beneficial biology in that layer hen manure, as well as the black soldier fly, without the risk of contaminating crops with pathogens, is something we work very hard on. So that's the product that we've developed. And also, as I've said, we've been working with Biome Makers to understand it for several years. So it's exciting to be able to share what we're doing.
So Will, it looks to me like you have a product that is actually, it has components in all 3 levels of our complexity ladder. You have nitrogen, phosphorus, potassium, calcium, and so on. You have more distinctly biologically active components like the chitin, say, and then you also have not just one or a few microbes, but a diverse assortment of microbes that are going to be delivered to the field with this product. So I say that to say, why did you choose to collaborate with Biome Makers on testing this product?
Yeah, great question. Scott, we know that there's a lot of good in layer hen manure. There's a lot of good in biology, but to your point with the piano key graph, what we have seen is that really understanding it and investing in understanding where this will be most impactful is going to be our best path to ensuring that we work with the right farmers in the right ways to make sure that they're getting the impact that we want to see. I think, frankly, we hate if our product does not work. And so being able to study this in depth with you all has been really the only path that we've seen as a way forward to be able to provide this with the backing of our company and our brand name out in the marketplace. So working with you all has been a tremendous process because it has allowed us to get really clear on what's happening in the soil. And frankly, being able to do that is also, I think, our best way as an industry to continue to get farm productivity high without necessarily over-relying on chemistry or some of the things that aren't working as well as they used to, like over-application of fertilizer. Does that make sense?
Absolutely. Okay, so biologically active products demands biologically relevant metrics. So real quick, if anyone is already, they think I'm well familiar with BeCrop trials, I have been in webinars about this before, I'd encourage you to hang on because we are adding a new layer of complexity to this story as we go on. But Jacob, first of all, will you give us a rundown of what BeCrop testing is, what BeCrop trials are, and what it is that we partnered with Chonex to do?
Yeah, sure. So we are very much aware, and we've known this for a long time, that soil is full of microorganisms. The difficulty is in being able to understand what those microorganisms are doing. So what we're able to do with biomakers, and this is something that has been done in large research institutions, it's been done in universities, but it hasn't been accessible to everyone. And the analysis, the interpretation of the data has often been a little bit murky. And so basically what we do is we extract all of the DNA from the soil. And here I've represented the DNA as circular. Usually bacterial DNA is circular, so I've just kind of represented it that way. And there are two ways that you can go about this. One way is that each one of these bacteria have a very short sequence of DNA that tells us exactly who it is. And this is kind of the gold standard that everybody uses to really classify what is the species. So if you send off your organism to a lab, they're going to use this little bit of DNA that they sequence. And that's all that we sequence. We just sequence that little bit. And so we get all of these little short sequences. And from that, we're able to go through and say, this is the list of all, you've got 1,000 species in your soil. This is the list of the name of all those species. That's the way that we do. So there are other groups that will sequence everything. They will go back to those original colored circles and they will sequence everything and give you everything. But the problem is that the sequencing is the most expensive part. And so, we're able to sequence a little tiny bit, and I'll talk a little bit about how we get the information out of this. So once we've got this list of 1,000 different species, what we're able to do is we're able to take it back to a phylogenetic tree. So that's what I've got on the right there. And what this tree is doing is we've been able to go through and we're able to say, okay, we know what this species does. We know what these groups of organisms do. We know that if we have Bacillus species 1, for example, we've actually sequenced that somewhere else. So we don't have to charge you for sequencing that because we have that in our database somewhere else. Or we've got something that is very, very, very closely related to that. And so in a lot of these cases, we're able to look and say, in this whole group, for example, we've got this pink group, and we're able to look at that pink group and we're able to say, every single one of the organisms in that pink group, they have the capability of making nitrogen available for the plant, whether that's through fixation or something else. But everything in there has that. So even if we find an organism that we've never sequenced before, if it falls within that group, we can reasonably assume that it has that characteristic as well, because we've never seen one that doesn't. And so based on that, we're able to then start to make some correlations and some ideas about functionally what is going on in that community. We're able to say, this is the proportion of the community that can do all of these different functions, and we divide that into these kind of larger agronomically relevant functions. So specifically, I'm actually going to work here from right to left. So we're able to look at the organisms that are responsible for cycling kind of the major nutrients that would benefit the plant. The way that we kind of look at this is we're not, we look at this as the capability of the organisms to cycle nutrients. So we're not looking at how much gas is in the tank. That would be looking at, we don't look at how much nitrogen is in the soil or how much gas is in the tank. We're looking at how fast that engine can go. And so we're looking at those cycling pathways. And we're looking to say, are you increasing that? Are you increasing the efficiency? Are you improving that? Or decreasing that in some cases. So we're able to look at these nutrient cycling pathways. We're also able to look at kind of phytohormone producers. These are the real key cycles that are responsible for these biostimulants. So when you have a biostimulant, you're either producing some kind of a stress adaptation for the plant or some kind of a phytohormone production. So we're able to kind of look at those two levels, but then we're also able to look kind of more broadly at biodiversity, functionality, resistance, biosustainability, kind of the big picture of saying how healthy is the soil, before and after you've treated it with something. And so that's where these trials come in. We have this we've got kind of a side-by-side where we've got this before input. So we know that the microbiome just changes naturally. So we usually are always going to have kind of not only a control where you don't treat it, but we also sample both the control, and if you go to kind of the next animated section, the after input, but we've sampled both of those before you've done anything to know kind of what is that baseline of microbiome. And then you'll treat one of those plots, and then we'll be able to look at the comparison of how they shift over time and with that treatment to be able to give you an idea of what exactly is it that your product is doing. How is it shifting those cycles? How is it shifting the organisms that are responsible for that plant health, for biosustainability? And so we're able to actually then quantify that and come back to you and say, this is what your product is doing in the field in different soils.
Very good. Okay, so chapter 1, Will, is you Chonex embarked on a number of trials across the Southeast. Can you tell us a bit about these?
That's right. So as part of our product development and really understanding our technology, we have launched, we're using CROs across the Southeast, trials in corn and cotton. These are from last year. And so what we're going to look at here is the results or some of the results of those trials. And we did BeCrop testing to understand the impact on soils, as well as tracking traditional yield increases. And so the BeCrop testing was quite insightful. So we're excited to talk about that. And we're going to zoom in on 2 locations. So one is Greenville, Mississippi, and the other is Chula, Georgia.
And there were 10 total trials, right? 4 sites had corn and cotton, and then there were separate sites that had just corn and just cotton.
Correct. And our agronomist, Charles Ed Snipes, has deep expertise, over 40 years of experience in this. So he's a very he's a stickler for the data. And so we are studying this in depth and what we think is to the highest quality we can. So, but yeah,
right, so Greenville and Chula. Let's talk about Greenville. What did we see?
So what was interesting is in Greenville, we're seeing a number of different functional groups of microbes, increasing in the soil. This is using BeCrop trials. So, these are 3 of them that we saw. So we saw plant growth hormone producers increased by 64%, over 100% increase in some of these groups that are known to build soil structure, exopolysaccharides. There's also heavy metals that we saw increased in Greenville, Mississippi, and this is using StrongSoil, pre and post using the BeCrop trials. And then what's interesting is you highlight Chula. We saw completely different microbial increases, which is interesting, and so very fascinating data that seems to be indicating that different microbial populations are being amplified based on differences in these soils. And so we saw yield increases in both cases, but the biological driver of those yield increases, to the point on the piano key graph, were different. And in Chula, what we're seeing is improvements in sulfur cycling, abscisic acid, which is for water use efficiency, and of course, a massive increase in salicylic acid, which is a stress tolerance process, which we'll talk about in a little bit. So,
yeah, and so for total clarity, for those who are really data heavy thinkers, this is percent change relative to the control. We have that control site, so we do a delta analysis looking at the change in these functional groups relative to where they're changing the control. So if it increased in the control, we actually had, say, over 125% increase in the treated, because these changes are measured relative to the control. So we saw here in Greenville, plant growth hormone producers, exopolysaccharide producers, and mitigators of heavy metal stress. And in Chula, we saw sulfur cycling, we saw producers of abscisic acid and producers of salicylic acid. Clear biological activity, clear product performance, but zero overlap. No rhyme, zero overlap whatsoever. So, Will, when your team was looking at these results, tell us about what was the thinking, the reaction when you're getting this data that is high resolution and it's in depth and it's clearly showing product performance, but not the same thing. Yeah,
well,
what was your thought process getting results like these?
Well, I think what it reflects is something that we all know, which is that, farms in Georgia are different from ones in Mississippi. Greenville, Mississippi is in the Delta. The soil structure is different there. If you go to Chula, Georgia, we're looking at different farms, different soil types. And so what we're seeing here, what we're excited about, is that we know that soils differ across the country in different regions. They therefore need and likely have different gaps in terms of their microbiome. And because of the complex, diverse nature of our product, what we're excited about is we're able to feed those different gaps or amplify that biology in different ways. And we've always thought about our product as more of a shotgun approach towards feeding the microbiome rather than a single, sort of rifle approach. And because we know that the soil microbiomes are very complicated, that's as exciting because we're able to cover more parts of the country and help some of those biological needs. So this is actually very exciting for us. But working with y'all and Biome Makers, we're able to really get deeper on understanding what exactly is happening. And that's been very important for us.
Okay, so give us a second to cook. I'm talking to the audience, not Will. So just looking at these, you do 10 trials. You can look at them sort of side by side, and you can try to unpack a pattern. Well, what we found was, all right, one commonality is the native microbiome composition was largely preserved. StrongSoil is not shoving or completely transforming the microbiome composition, the bacteria and fungi present in hugely disturbing ways. I use disturbance in the scientific sense, which is a positive thing, because when you shift the native microbiome a big distance from where it comfortably is, then it will inevitably shift back. The sorts of inputs that you see a huge composition change from are, you would consider them disturbances. They'd be biocides, they'd be high salt index fertilizers. In terms of nutrient cycling, we saw moderate and variable benefits. We saw increases in nitrogen cycling in the corn trials in particular at 8 out of 10 time points, 8 out of 10 samplings. We saw increases here and there at different sites of mobilizers of calcium, magnesium, copper, and chloride, but not 100% consistent. We're seeing a piano graph in those. In cotton, nitrifiers increased at 8 out of 10 sampling times. So we have some commonalities there that you'd have at least something of a reliable pattern to in the Southeast. In stress adaptation, there were frequently massive and remarkably variable benefits, as we've highlighted with Greenville and Chula, where you'd have changes in abundance relative to the control ranging from 63% to 760% in producers of salicylic acid, abscisic acid, heavy metal tolerant, plant growth promoting microbes, and exopolysaccharide producers. And Will's putting a great face on it, but I remember when we were first looking at these trials in isolation, especially the sales team were kind of scratching their heads and going, what do I do with this when I want to sell in Texas? What do I do with this when I want to sell in Louisiana? Like, what expectations can I actually give based on this data to the farmer or to the potential distributor? And so this is where the plot thickens, because at the same time that these trials were going on, our team was working on BeCrop Farm, which, if you don't know about it, basically we had to find a way to make our soil genomic testing, we had to make our biofunctional testing accessible and affordable to farmers, crop advisors, and retailers in such a way that we could capture biological function across an entire field without breaking the bank. And one of the strategies that our team took, one of the main strategies we took, was we had to learn how to integrate environmental data, remote sensing data, in order to both target our samples and generalize from our sample points to the surrounding areas. Well, as we're working on this on the one hand, and we're getting these very variable results back for the Chonex trials on the other hand, it occurred to us the same technologies, the same tools that we have been developing and integrating with BeCrop Technology to produce BeCrop Farm, we can take these same factors that inform microbiological function and use them to do a meta-analysis of the results of these different sites and understand, well, begin to investigate what environmental factors are playing a role in explaining the variability, in affording the opportunity for strong soil to behave this way or that way, in affording these different results. So part of that is, okay, what factors that influence microbiome composition and function can we assess remotely? It's great if you can get the CRO to collaborate and to give you, your CEC and your base saturation, your organic carbon for each plot. And in the future, and as we are now with Chonex, we are planning future trials around these capacities. But at the time, we knew that, okay, we can get a lot about temperature. You can actually remotely sense soil moisture in the upper layers. We can get solar radiation. We can get some data about the soil parent material. We can get data about annual precipitation, variability in precipitation. There's a whole lot of environmental parameters that we can lay over the top of multi-site trial data and start to look for relationships. And so our team had already identified for BeCrop Farm different environmental zones where we anticipate, based on environmental factors, distinct microbiological functions, okay, distinct drivers, distinct selective pressures on the microbiome, on crop health. And so, even though all these trials were in the Southeast, we put our heads together with Chonex, and we decided that we would do a first of its kind, for Biome Makers at least, meta-analysis across these sites. And it is to this that I will hand over the floor now to Dr. Jacob to dig into the nitty-gritty that the first half hour has been prelude to. Bless you all for your patience and attention.
Yeah, so here you can see this map. It's the same map that Scott just showed, and we've highlighted kind of the locations, and you can see that we've got the locations in 2 of our different zones. So we had predicted beforehand that the biology in these 2 areas is going to be different. And what we've got below are NMDS plots. And so essentially what it is we take all of the information on either the bacteria or the fungi. So all of the microbiome, who is there, how abundant they are, and we compress that into one point. And so the axes don't have any dimensions, and kind of the most important thing to understand is how close they are to one another. So the closer they are to each other, the more similar those communities are going to be. And so we've got it divided into 2 different axes. And so again, they don't mean anything, but really kind of what it's saying is there are 2 dimensions that we're really going to be looking at. The interesting thing, and I've kind of circled this, is, for example, if we look at bacteria, we can see that on that first axis, Bainbridge and Chula are on that same line, right? And so the one component that explains 27.4% of all of the variation is almost identical in those two. And so we kind of have this clustering on that axis of the Georgia samples, and then everything else is clustered together, which kind of fits with what we had predicted would happen, that we had two different zones. And we can see that same thing on that second axis with the fungal composition, where again, Bainbridge and Chula are kind of separated from those other four in that second dimension. So it kind of confirms what we had predicted, but there's quite a bit more that we can get from this. So really, hop back for just one more second. I want to show one more thing about this, and that's that. So the gray are the cotton trials, and the green are the corn trials. The triangles are going to represent basically the different treatments. And so one thing that we wanted to point out is that first of all, the crop is what's shifting it. Especially if you look at Chula, you can see that the greens, all of the green are off to one side, all the gray are off to another side. And so what this is telling us is that the crop is having the dominant impact, but it's also saying that the Chonex product, the StrongSoil, is not something that it's like, well, it only works for cotton. It doesn't do anything for corn. So we're not seeing any indication of that, and that's basically what we're seeing here. We're seeing that the location, we've highlighted that in the table. Location is very different between these. The crop is playing a big difference, but when we look at the treatment itself is not shifting the microbiome in one direction or another direction, and so it's able to work for corn as well as for cotton. So now if we go to the next, this is kind of a different way of looking at that same thing, where we've got kind of the two different zones. But we've also then looked at, this is just looking at the metadata that we have. So this is trying to explain without so much the biology, what is it that's driving the difference in these fields. And so we have the biological side, which are the BeCrop indices, but then we also have the environment and trial design associations that I kind of want to focus on here, because we can see that in the purple, which we've kind of got shifted off to that one side, we've got higher soil temperature, and that's in the Bainbridge and Chula. And when we look at that, we kind of say, okay, this is really what is distinguishing these from an environmental perspective. What is making these soils different environmentally? As we look at all of the statistics, as we try to figure out what is really, pushing these soils to be different, it is higher soil temperature. That's what's, I'm going to call it, that's what's stressing these soils to be different than everything else. That is what's driving them to be different. And as we start to kind of then pull out the biology of what is happening in the soils, not necessarily what's happening in those soils, but what StrongSoil is doing in those soils, we can see that it's that stress adaptation of salicylic acid. So we've got, in this case, we've got all of the trials. So the cotton as well as the corn. The corn is there kind of in the middle, and the cotton is off to the right. But you can see that in every single one of these cases, often at both time points, we're seeing this increase in salicylic acid, over 100% in some cases. And when we look at what salicylic acid is able to do, we pull up this paper that talks about how salicylic acid is able to help ameliorate heat stress. And so, in this case, we're able to say, well, maybe what's happening is these soils are experiencing a lot of heat, and in the case of these soils, what Chonex is doing is, or what the StrongSoil is doing, is it's really increasing those parameters that are associated with that heat tolerance.
So environmental stressor, I'm in the wrong slide. We're misordered. Beg your pardon. Hold on. Here we go. Key environmental stressor, higher soil temperature. Specific microbial adaptation, amplification, salicylic acid producers. They ameliorate heat stress as well as other abiotic stresses. They ameliorate those damages and enhance photosynthetic efficiency. All right, let's look at our other location here.
Yeah, so the other location, we wanted to focus on this, Greenville, the evapotranspiration. Now this is saying that this is what is driving this community to be different, is the amount of evapotranspiration. So to me, this is suggesting that there's some stress that's associated with evapotranspiration.
Mm-hmm.
And now when we look at, okay, well, what is StrongSoil doing in this soil? I think we did see some salicylic acid, but it wasn't as strong as those other cases. In this case, the thing that stood out was about our exopolysaccharide production.
Mm-hmm.
And so you can see that we've got 60 to 100+% increase in exopolysaccharide production from the StrongSoil component of this. And so evapotranspiration is often associated with kind of this water stress, drought stress, and then the exopolysaccharide producing bacteria are there to again ameliorate this stress, which is through making it so that the water is more available to the plant. So what we're seeing is that in soils where you have heat stress, adding StrongSoil basically improves the organisms that are responsible for helping the plant with heat stress. Where you have drought stress, adding StrongSoil improves those cycles, the pathways, the microorganisms that are responsible for helping plants with drought stress.
So if I may attempt to articulate this another way, out of all the sites, the environmental parameter most distinctive of Greenville was the combination of solar radiation, relative humidity, wind, air temperature, vapor pressure deficit that leads to greater evapotranspiration in crops generally. And that is an indicator of water demand. Would that be accurate?
Yes, and that's what's really stressing out not only the community, but all of the environmental factors as well. So that's what is distinguishing those soils above everything else that we looked at. All of the other parameters, soil temperature, moisture, all of these things. This is what is really saying this is what is making these soils different
So
most.
there's a rational relationship between the most salient stressor in Greenville, Mississippi, and the biological function that we saw upregulated where StrongSoil was applied.
Correct.
Yes?
Yes.
And so here's our going backwards, out of order slide. Key stressor, evapotranspiration, i.e. indicator of water demand. Specific microbial function amplified, exopolysaccharide producers. They help develop soil structure. They help retain water. They help retain nutrients. They help build that protective rhizosheath around the root environment in the rhizosphere. Okay, so we have a data-informed hypothesis now from this meta-analysis. This meta-analysis that, mind you, was derived from trials, not optimized for such an analysis. It didn't cover that many of the distinct environmental zones that our team identified. We were able to get, based on trial design, less data pairings between BeCrop and environment than we might have otherwise had we designed for this, because we didn't know when planning the trials together with Chonex that this was going to be possible. Nevertheless, this meta-analysis gives us a data-informed hypothesis that StrongSoil potentiates selective microbiome recruitment to mitigate the salient limiting stressor at these different sites, particularly in the Georgia cluster and in the Greenville location. And so now, Will, tell us about this year's trials and the way that we are keeping these capacities in mind moving forward to this current season.
Yeah, happy to. I mean, we are continuing to do these BeCrop analyses with our current trials. We frankly believe that we need to continue to repeat these studies to understand if they're replicable. We have a strong belief that they will be, but we want to keep seeing this pattern emerging. So that's one thing that we're continuing to do. So we love our partnership with Biome Makers and with our current trials. That's what we will be doing. We're also expanding to other crop types to understand the impact that amplification of the microbiome can have in things like citrus or berries or vegetables. So we're excited about continuing to research and study this. But as we see these patterns emerging and these zones emerging, and we can really understand those, we're pretty excited about the potential impact of how StrongSoil can be kind of a single, we almost are thinking about it like a multivitamin, where we can go into your field and fill the gap that might be needed based on the stressor relevant to those zones. So that's very exciting for us. And data like this, frankly, going back to that piano key graph, it's also very exciting because that means that we're more likely to be able to help farmers go more towards the right in that graph with confidence, and that's ultimately where we want to be.
Mm-hmm, mm-hmm. And as I understand, branching into 3 new states as well, and peanuts.
Yes, we
and states on the outer edges of where you've tested before, and so you're casting a wider net to better understand these environmental indicators.
What we've been able to do, and frankly, and this is sort of gets back to the kind of experimental, ingenious nature of farmers, is we've had a lot of farmers who sort of give us a shot and started to get some really good results. So we've done some great results in blueberries and peanuts. We haven't done as many trials there, but those initial shots on goal that have shown that we get some really good lift is suggesting to us that there's something really good happening with when you combine StrongSoil with these different crop types in these different farms. And so being able to use that as an observation that then we can go back and start to study working with Biome Makers has been our approach all along. So yeah, the peanut trials, blueberries, citrus, we've gotten some great results there, but have not done the hundreds of trials that we've done, in corn, cotton, and soybeans in the same way. So we're excited to keep doing that, and we plan to keep doing that every year, as long as they'll let us.
Excellent. We are honored by your collaboration. And so, call to action, input folks. If you have a biologically active product, incorporate biologically relevant metrics into your trials. And I would urge you to get in a dialogue with us about designing those trials, optimizing those trials for a multi-site meta-analysis like this. And for those who are distributing, selling, or using biological products, first of all, hold your product developers accountable for knowing the how and the when and the why and the where their product works, and then test your own fields. I mean, circling back to BeCrop Farm, there's other webinars about that if you want to understand it better. But does field X have a need that fits the performance of that product? And do your conditions match that? Now, we've been building that kind of data into the AI recommendation system that's built on all of the trials data from our collaborations over the last decade. But the potential of all this, if our typical product performance is variability as liability, if you can understand what is driving performance, what is driving that variability, if you can understand not just, okay, Greenfield, Tennessee, it seems like maybe we had a nothing burger, but why? What was it? Were the stressors that drive performance absent? What factors might have been at play? ultimately the goal is to eliminate the left side of this chart and turn the variability, the pluripotency of biologically active products into an asset through the insights, through biologically relevant insights, so that your chart looks more like this. There's soil microbiome, soil ecology. There's always going to be stuff that is beyond our accounting. There's too much emergent, synergistic. There's always going to be components that are mystery. But with biologically relevant insights, there's no reason why you shouldn't be able to push your graph toward a higher win rate. That's Lucia. I'm hoping that we can stay past the hour a little bit to answer questions because we took 50 minutes to tell our story, but thank you all for listening. And now I'm going to pull up the questions that I'm being sent and pose them, to my co-panelists. Okay, so here's a question for you, Will. Interesting formula in StrongSoil. How was the soil/plant productivity mathematical formula arrived at? You can tell me if that question makes sense to you. How was the soil plant productivity mathematical formula arrived at?
This is for me.
Yes.
Well, the thought here is that we have multiple factors impacting farm productivity, whether that's nutrient availability, water, sunlight, obviously agronomic practices. So all of these things are kind of in conjunction, and I couldn't tell you the exact formula, but there's a number of different factors that are impacting farm productivity. And I think what we have identified, again, and Biome Makers has been a great partner in this, is that the microbiome of soils, as well as plants, leaves, etc., also must be maximized or working in conjunction with all of those other factors to be able to maximize the productivity of a farm. And so what I think historically we have not had as much insight into the microbial health of soils to understand that its impact on farm productivity. We've had a lot of great tests that have shown, whether or not we have enough chemistry, whether or not we have pest issues or disease issues. But what we believe is that we also have to look at improving the microbiome of soils to be able to continue to maximize the productivity of farms and do so in a way where we're more efficient with some of the other factors that we have perhaps been over applying, such as fertilizer, etc. So did that answer the question? I hope. I'll write the formula out on a piece of paper, on a cocktail napkin, and take a picture.
Can you tell us about why you chose, why Chonex chose those particular trial locations?
Yeah, so part of it is we are an Alabama-based company. So you'll notice that we've got southeastern roots. We're very keen to focus on helping. Ultimately, our founding mission and reason for being was because we have seen farmers struggling in the Southeast, in Alabama in particular. And so we have developed deep roots here. And so a lot of our connections and activity has been focused in this area. But we are excited now to be able to do trials in additional states. We are actually looking to enter into the leafy green market in Arizona and California. So looking at doing trials there. And so going national has been something that's coming. But ultimately, we've started in our backyard. So that's why we're looking at Mississippi, Georgia, Alabama, etc.
Mm-hmm. One question asks, are the functional groups measured at different locations based on the microbial population dynamics, i.e. based on 16S sequencing? The answer is yes, in part. 16S sequencing for the bacteria and archaea. ITS1 gene sequencing is the gold standard marker gene for fungi. Now, Dr. Jacob, expanding on that question, someone asks, it's well known that metagenomic sequencing can underrepresent fungal communities, as fungal DNA is often overshadowed by bacterial sequences. How does Biome Makers address this challenge in their sequencing approach to ensure accurate detection and representation of fungal taxa?
Yeah, that's a great question. So we've got some slightly different markers that, some different primers that we use to look at them. Again, it doesn't ensure that we're not getting it, but one of the things that we do in order to try to overcome this, or maybe not even overcome it, but account for this, is when we look at these quantifying and comparing these, we're basically comparing fungal to fungal communities. So we're not necessarily saying this is exactly the number that is there. What we're doing is saying, when we can detect this much, this is what it means. And so by doing that, we're not saying that we are getting a full view of the fungal community. I think there would be better ways of doing that. For example, the way that we discussed before about sequencing everything would be a better approach to really understanding exactly what the full fungal community is and what everything, all of the different sequences. But what we're trying to do is we're trying to get a high level picture. And so in that case, what we're able to do is we're able to say, for example, in the control, are we seeing more fungal or less fungal, whatever cycling pathways that we're looking for. And so in that way, we're able to get kind of that relative, and we're able to look at that, and we're able to compare that. So if you wanted to say, I want to do a full fungal consensus of this community, you probably wouldn't do our approach. You would do a full shotgun metagenomic sequencing to really drill down and look at what the fungal community is.
May I actually take a stab at that and get your reaction to it? So the question pointed out that it's metagenomic sequencing that underrepresents the fungal community. And I think that makes perfect sense, because in agricultural soils, you almost always have bacterial dominance. And so you're going to have far more instances of bacterial DNA sequences than fungal. The fungal is in the minority. Well, because we use an ITS1 marker gene that targets and will only hybridize with fungal DNA, then unless you're going to spend heaps of money to exhaustively run a sample, then amplicon sequencing, to my money, would be the way to go, because the metagenomic companies out there, they don't sequence all the DNA in the sample. It's prohibitively expensive to do so, 5 or 6 times more expensive than amplicon sequencing. Am I wrong about this in thinking that you might get a better representation of your fungal community by targeting it with a marker gene that only hybridizes with those sequences?
Yeah, I mean, it really comes down to the question you're asking about why, like, what do you want to get out of the fungal community? If you want to get kind of a big picture of what's going on, kind of at the level that we've been discussing, then yeah, Scott, you're right. An ITS would be fine. If you want to get down and say, well, I want to see how underrepresented it is, then yeah, that's like a different question, and that's not really a question I think that a farmer would be asking. That would be a more research question, and then you kind of say, well, okay, why do you want to look at that? And so it depends on the question that you're asking. As far as getting out into the field, something like what we've been discussing here, of saying we want to see big picture functionality, functional shifts, this works. If you have a different question, then you might need a different approach.
Okay, thank you. That is very good. We got far more questions we got many great questions, and it was good that we took the requisite time to tell the story well. But if you have asked questions that we were not able to answer, I'd love to do the Joe Rogan experience and hang on for another 2 hours to answer them all. But Dr. Jacob and I have an internal meeting that's starting in 1 minute, so we'll have to go. If you ask questions that weren't answered in this call, in this webinar, you will get an answer by email. We will be in touch, and be sure that you get your answer. If it's a Chonex related question, I will pass it to Will, and otherwise Dr. Jacob and I and other members of our team will be in touch with you. We thank you all very much for your time and attention. We don't take it lightly. there's a lot of other things you could be doing. So peace to y'all and yours.
Thank you. Take care.